introduction, Jen. As you said, I'm Dr. Milligan-Myhre. I'm at the University of Alaska Anchorage. I included my Twitter handle here and my website, my lab website. So if you're interested in learning more about this after the talk, you can check those out. Today I'm going to be telling you about adapting an evolution model for host microbe studies, and specifically I'll be talking about the fish stickleback, three-spined stickleback that we work with here at the University of Alaska Anchorage. And it's located here in Alaska and throughout the Northern Hemisphere. So I wanted to tell you a little bit about my background, because at the very end I'm going to be switching over to some pathogen stuff, and I wanted to explain why I'm going to do that. I've been interested in host microbe interactions since an undergrad, but as an undergrad I actually started out researching pathogenic microbes, or microbes that can cause disease. And as an undergrad and for 3 years as a lab tech, I studied the gram-positive bacteria, Staphylococcus aureus, and I identified environmental and genetic factors that regulate toxin production of Staph aureus. And then as a graduate student, I switched over to Toxoplasma gondii. This is an obligate intracellular parasite. And so here you can see the parasites, and they're inside of a host cell. And here we've stained them with pretty colors. And we injected mice with these, and then determined which genes were necessary for the parasite to make it from the site of infection to the brain to establish chronic infections or to affect acute infections. And I did that at the University of Wisconsin, Madison, under Laura Knoll for my PhD thesis. So my background really was in pathogenic microbes, and I switched over to beneficial microbes when I became a postdoc at the University of the University of Oregon. Right now I'm at the University of Alaska Anchorage, and before I get too deep into my talk, I wanted to tell you about the people who've done this work. So my lab has a lot of undergrads. We have Coral and Kelly who are working on assays for the immune response to microbes, and I'll talk about that in a minute. I have Kenny Sparks, who is also an undergrad, who is working on mock communities and determining how antibiotics affect how the host genetic background affects antibiotic disruption of the microbiota. Ruth Eisenberg is a lab tech who's worked on all the projects, who just left to start grad school at my alma mater, University of Wisconsin-Madison. I have a postdoc who started with me, Emily Luskak, and we'll be talking a lot about her work, and a master's student, Ryan Lucas, who's working with Emily on the antibiotic projects and comparing the microbiota in the water to the microbiota in guts. Tim Stevenson helps us with our bioinformatic works. Jeremiah Lewis and Rachel Cramp just started, and they are looking at microbes in the environment and microbes in the gut, and isolating and characterizing different microbes. So what is our lab focusing on? Why do I have all these people? We have this really big question of how does the host genetic background contribute to host microbe interactions? And the reason that we're studying this is because we know that the microbiota are actual communities of microbes that live within a host. It's not just one type of microbe that the host has to respond to. And we tend to think of this in humans, right? Because we're very human-centric people. We know that microbiota plays a role in host health. We know that it stimulates development of certain tissue types and cell types. It prevents colonization by pathogens, amongst a lot of other things. We're really interested in the microbes in the gut because they play such an important role in helping us digest our food and recruiting stimulating the immune system. And it stimulates the immune system by both recruiting innate immune cells, including neutrophils and other immune cells, and also helping the immune system develop. So here I'm showing a stain of some different immune cells that will be found in the blood. We're specifically interested in neutrophils. Neutrophils are an innate immune cell that responds very quickly to a site of infection, but they're also naturally circulating within our body. And one of the things I'll show you in just a minute is that neutrophils are always present in the gut. Low numbers when there's not an infection, and their numbers increase when there is an infection. And their role is to be there right away to consume pathogens and make sure that the host is able to clear those pathogens. Now, there are multiple factors involved in the disruption of host-microbe interactions. So we have the microbiota in the gut, which we see here, that gets disrupted in patients who have inflammatory bowel disease or other diseases of the gut. But then we also have an immune response to microbes in the gut. Here we have a healthy gut, an H&E stain of a section of a healthy gut versus one with ulcerative colitis. And this is showing that these little dark purple dots are immune cells that lead to inflammation of the gut and distortion of the gut physiology. And we know that there's this balance between the microbes that are in the gut and that community of microbes, not just one type of microbe, but communities of microbes that change the way that those microbes interact with the host, and this immune response. But in addition to this, in the case of inflammatory bowel disease and many other inflammatory diseases that affect the microbiota, we know that there are a large number of genes that are affected that influence this balance between the immune response and the microbiota. In other organisms, if we were to study the role of these genes or to try to identify which genes are really important for these phenotypes, we'd have to do a long process of knocking out one gene or disrupting one gene and then disrupting another and then disrupting another and then disrupting another. And that would be in organisms like inbred mice or inbred fish that have been used for a long time in host microbe interaction studies. So since we're interested in the host genetic background and its role between the microbiota and the immune response, we decided to use a model that actually has natural genetic variation. So something that would already have genes, a number of different genes that influence that relationship that are different between one host and another, and specifically between one population and another. And so we decided to use this fish, Threespined stickleback. This fish is actually characterized by these spines. That's why it's called three spines. They have three spines here. They've long been used as a model for host microbe interactions. They've long been used as an evolutionary model, and I'll talk about that in just a minute. But one of the reasons that we wanted to use this fish is because in the past, when host microbe studies were first started in gnotobiotic animals, and here I'm showing some pictures from the 50s of host microbe interaction studies in which animals were made germ-free, and you can see that back in the 50s we were using models that are not standard now. So rabbits and mice that actually were not inbred. We've gone away from these animals that have natural genetic variation, and we've started using inbred animals, and that makes sense if you're trying to control for genetic background. But if you're trying to determine which genes are responsible for these interactions, or if you're trying to determine the relative role of different genes in the interactions between the host and microbes, then it makes sense to use an animal model that has some genetic variation. And so as a postdoc, I joined Karen Gilliman's lab. She's interested in host microbe interactions, and we started a collaboration with Bill Cresco, who had a lot of background in using determining the role of the host genetic background in different phenotypes. And we developed this model for Threespined stickleback to use Karen's expertise in host microbe interactions and Bill's expertise in host genetic background influences to determine how the host genetic background contributes to host microbe interactions. And the reason that we're using Threespined stickleback is because they have an awful lot of natural genetic variation. But there are other reasons, and I'll get to that a little bit more in just a minute, but there are other reasons for using stickleback. We get large clutches from single crosses. So for example, if you take one female fish and cross it with one male fish, that female fish can lay up to 200 eggs, sometimes even more. So then we have this huge clutch of eggs from a single fish, and each of those eggs will have some genetic, will be siblings of each other. So this allows for these really big experiments where we have lots of numbers of samples. The juveniles are optically transparent. So here I'm showing some eggs developing into a fish. This is hours post-fertilization. So here's an unfertilized egg, and you can actually watch the development of the fish inside of that egg and through the juvenile stages. So here's 8 to 9 days post-fertilization. This is a transparent fish. And one of the things that we can do is we can actually watch microbes live as they interact with these fish inside the fish. The evolution and development of stickleback have been thoroughly described. So this actually comes from a paper in which the development of stickleback has been described. So we have a nice model where a lot of the development is actually known. Importantly, sticklebacks share many pathways that are involved in host microbe interactions and immune response to mammals, including genetic determinants of the immune response. So for example, we have neutrophils and B cells and T cells in stickleback, and we have many genes that regulate these that are similar to humans. And other people, including the Kingsley lab, have used this to determine genetic regulators of things, for example, bone development in stickleback. So this is showing a marine phenotype that's long been used in stickleback to study the difference between marine fish and freshwater fish. And here you can see this marine fish has a lot of bones, these bony plates, and the freshwater fish don't have very many of these bony plates. The Kingsley lab identified a gain of enhancer that is lacking in the freshwater fish, and found that this gene, this regulator, is actually conserved from humans, from fish all the way to humans, and that loss of this enhancer in humans resulted in human-like feet compared to chimp-like feet. So this thing that we found at the Kingsley's lab, this regulator of genes that was identified in stickleback, actually is conserved all the way through humans. And so by examining something in fish, we were able to identify something that was really important for the evolution of humans. Threespined stickleback also have a lot of natural genetic variation. This is not an inbred model. This is a model that has been used throughout evolutionary studies for over 100 years. And also stickleback, and so we'll be using this, we'll be taking advantage of this natural genetic variation to identify genes that are responsible for host microbe interactions and find out which genes are really important for these host microbe interactions. And finally, the gut morphology is really similar between between fish and humans. So in other fish models, other fish models don't have a true stomach, or their gut is not as doesn't look as structurally similar to humans as stickleback. In stickleback, we have an actual esophagus and a stomach with gastric pits, so a lower pH that's similar to humans. And then we have a gut that is divided into at least 2 distinct parts, and we think it might actually be have 3 distinct parts similar to human guts, and it has folds just like human guts, and a lot of structure. And this is at 13 days post-fertilization. So this gut actually, and this is a juvenile fish, so this gut actually has structure that looks similar to humans very early in development. This also gives you an idea for the size of fish. So mostly I'll be talking about juvenile fish, and juvenile fish are about 5 millimeters long at 13 to 14 days post-fertilization. And as adults, they grow up to be about a couple of inches. So I keep talking about stickleback being this evolutionary model. Why did it become an evolutionary model? So here I'm showing the state of Alaska, which happens to be where I'm from. Here's Anchorage, where we're located. I'm from Kotzebue, Alaska, located up here. In Alaska and throughout the northern hemisphere, there have been populations of stickleback that originated in the ocean, but have been isolated in different bodies of water. So I really like this map because the bodies of water are really distinct. There are these blue blobs throughout this map. Anchorage is down here. This is a road that leads to several of our populations. And you can see that throughout just our little area around Anchorage, there are a number of different bodies of water. Stickleback from the ocean have either been established in these bodies of water recently, sometimes as early as like 5 or 6 years ago, or became isolated in these bodies of water as glaciers receded. So for example, these are some nice chains of lakes that were formed when glaciers receded. And as those glaciers receded, populations of stickleback were isolated in these different lakes and now have been evolving in isolation from each other and in isolation from the anadromous or oceanic population for tens of thousands of years. And so now we have repeated evolution experiments in which we can compare the differences in the host genetic background and host microbe interactions in these different populations. Now we can do these host microbe interaction studies in stickleback because we're able to generate notobiotic stickleback. So what do I mean by notobiotic? Noto refers to known, and biotic refers to life. So what we're doing is we are manipulating what life is associated, microbial life specifically, is associated with these stickleback. So here we have our conventional stickleback. It has lots of microbes. And for those of you non-microbiologists, these microbes are not drawn to scale, I promise. But I'm just showing a diverse community of microbes that are associated with this particular stickleback. We can take the eggs from this conventional stickleback and treat them with soaks of PVPI and bleach and grow these in a sterile environment. So the PVPI and the bleach kills off the microbes from the surface of the eggs, but doesn't affect the development of the fish. And then we can grow these fish in sterile water in flasks. The fish will become their guts will open at about 12 days post-fertilization. We tend to add microbes back to the water if we're looking at conventional fish at 10 days post-fertilization. And then we can raise these fish without food up to 14 days post-fertilization. I like showing people what the process looks like. So this is what our hood looks like when we're actually making germ-free fish. And we can raise the fish in these flasks that you see here. And we can either raise them germ-free, or we can add back microbes, as I said, at 10 days post-fertilization. And in a little bit, I'll talk about the collection of microbes that we have from stickleback guts. And because we have that collection, we can also add back individual microbes for a monoassociation, or we can add back combinations of microbes. And as I said, Kenny's creating a mock community where he has 20 different gut microbes that we're adding back to germ-free fish. We have many tools to study host microbe interactions in stickleback that we've adapted from zebrafish and from mouse studies. We have a stain for those neutrophils that I was talking about, those innate immune cells. So here's a cross-section of a fish. We've fixed the fish and stained the neutrophils with a dye that binds to the myeloperoxidase enzyme that's found in neutrophils. And then we do a cross-section of the fish. We cut it in thin slices in this direction, and then we can look at those sections under a microscope and count the number of neutrophils in each section and count how many neutrophils are in the entire gut of the fish. We can look at the microbial community with 16S pyrosequencing, which I'm sure many of you are familiar with at this point, but it's just sequencing this gene that's found in bacteria. It's also similar to the mitochondrial gene, but we've messed with the primers that we use to be able to identify bacterial 16S genes as opposed to stickleback 16S genes. And we can use primers to the conserved regions to sequence through variable regions seen in these little colored areas. And that gives us a way to identify one type of microbe from another in a large community of guts. So we can identify which microbes are there. And then we can also isolate live microbes. So we take a gut from a fish, we homogenize it, and isolate live microbes. I was trained as a microbiologist, so I really love playing with these microbes, identifying them, characterizing them, looking at biofilm growth and motility and all the other fun things that you can do with microbes. And then, as I said, we can add them back individually. And then we have protocols for isolating RNA and DNA to compare the transcriptome and the genetic background of the host and the gut microbes, the microbes that are associated with the gut of those fish in a single sample. So, my lab, that's all the intro. So what is my lab actually working on now? So we're interested in how the host genetic background contributes to host microbe interactions using this really great model that's been used in evolution for a long time. The questions that we're tackling right now are, how does the host genetic background contribute to microbiota membership, contribute to the immune response to microbes, and how does the host genetic background, or does the host genetic background, protect against disruption of the microbiota by antibiotics? So we're going to start by talking about whether the host genetic background contributes to microbiota membership. First thing that we wanted to do was to look at which microbes are actually in the gut. So this is a figure from Rawls et al. In 2006, in which they compared the microbes that are present in zebrafish to those found in mouse to those found in humans. Because, of course, one of the reasons that we're interested in this is to identify genes that are involved in host microbe interactions in humans. So are there microbes that are present in stickleback that are present in humans? Yes, there are a bunch. There are also stickleback have microbes that are present in zebrafish, mouse, and humans. And we also have some microbes that are present in stickleback that are found in zebrafish, but not mouse and human. And we have some microbes that are present in stickleback that are present in humans that were not found in zebrafish. So we think this is a gut microbial community that has members that are similar to both other animal models that have been used for human studies and similar to humans, just in which microbes are present. Now, one of the things we were interested in was when we started looking at which microbes are present in the gut, one of the things we're interested in is variation from one host to another, from one individual to another, because we know that in humans, we have natural genetic variation in our human populations. Do we see that in stickleback? And yes, we do. So this is a bar chart showing the different families of microbes that are present in wild-caught Oregon fish. At the time, I was a postdoc in Oregon, so these are fish that were collected off the Oregon coast. And what we can see here is that each of these is an individual fish, and each color represents a different family of microbes in these individual fish. So we see that there is variation from one host to another, just as we see variation from one human host to another. We wanted to determine if there are differences in the gut microbiota between oceanic and freshwater fish. And this would reflect the differences in the populations, human populations. We know, one thing I forgot to mention earlier, is that we know that stickleback have genetic variation that's actually from one population to another that's similar to the genetic variations observed in humans from one population to another. So we wanted to determine whether our population of oceanic fish would have a different microbiota from freshwater fish, which would be a different population. So again, we were in Oregon, and we did see differences in the gut microbiota from one population to another. Again, these are individuals. This is at the family level. Each color represents a different family level of microbes. And we can see that the microbiota in freshwater fish look very different from the microbiota in oceanic fish. They have some similar members. So for example, this green bar is here, and it's also in this population. And this brown bar is here, and it's in this population, etc. But we see that there's a lot more variation. And And in fact, when we look at family richness and family evenness, two different alpha diversity measurements, we see that the wild Oregon oceanic population has less diversity than the freshwater population. So we have a difference in which microbes are present, and we also have a difference in the diversity of these microbes. One of the things that this doesn't show is that we had differences in individual members of the microbiota. So for example, tourniquets were only found in the marine fish, but they were not found in the freshwater fish. And the Vibrio were much higher in oceanic fish. Now these are all wild caught, so there's variation in the environment that these fish are in and that their microbes are in. So we didn't test the microbes in the water, but most likely these fish are developing in the presence of very different microbial communities, and that may explain the differences that we see here. So one of the things that we're doing now at the University of Alaska Anchorage is we're comparing the microbes in the water to the microbes in the gut of these fish. And we're doing this to determine whether different populations of stickleback are selecting for different microbes from the water. And this also gives us an indication of what type of microbes these fish are developing in. So for example, if we have fish from Lake A and it has a different microbial community from fish in Lake B, both in the water and in the gut, we can start to piece apart whether or not those differences are strictly due to the different microbial communities in the water or whether they're due to selection by the fish. And this is a project that we started last year. We've collected fish from the bodies of water seen here. This is Anchorage. Here we go. This is Anchorage. University of Alaska Anchorage is right here. One of the sites that we collect from is Cheney Lake, which is about 5 blocks away. And then we collect from Mud Lake, Big Lake, out in the valley. This is about an hour. Big Lake's about an hour and 15 minutes away, just to give you an idea of how close these populations are to us, so we can collect on a regular basis. And we also collect from Westchester Lagoon, which is right here. And that's something that's just a 15-minute drive away, so we collect from here quite a bit. And then we also go out into Talkeetna and collect from a series of lakes that are isolated from each other but have some water exchange. So for example, Christensen Lake is separated from these other lakes by a road, and so there's not much exchange of water from and microbial communities from Christensen Lake and these other lakes, but there is some water exchange between X, Y, and Z Lake when these areas become flooded, and that's something that we're confirming. And we are also trying to determine whether these stickleback populations are also being moved around from between these different lakes. Now, I worked at a bench for 20 years, and I just started getting into field work, so I like showing that we actually do field work. And this is an exciting part about being at the University of Alaska Anchorage and being able to drive out to Westchester or Cheney on a regular basis to collect fish. So this is just showing my postdoc Emily, undergrad Coral, and a high school student Gary that was working with us for a summer, collecting fish at Westchester Lagoon. And here we are pulling traps from Cheney Lake. We're actually doing a longitudinal study in some of these bodies of water, including Cheney Lake, and this is Ruth collecting from Cheney Lake. And then we pair that up with our bench work. So we have field work and bench work. And here we are just isolating microbes from the gut. So we knew that we had these we have these samples that we collect from the wild, and we wanted to determine, what we see in the wild, can we actually recapitulate that in the lab? And so we brought some fish from the wild I grabbed some fish from the wild, some adults from Westchester Lagoon, and we looked at their gut microbiota and compared those to juveniles that we raised in the lab. And here what we're doing is we're just trying to recapitulate the gut microbiota from the wild in the laboratory. And here we have our adult wild guts and the microbial communities in those adult wild guts. And here we have some juveniles that were raised in the lab in water that we seeded with water from Westchester Lagoon. And what you can see here is that the microbes that are present in the wild we're able to establish in our lab-raised environment. And this is also giving you an idea of the different types of microbes that we have in our wild population. Some proteobacteria, which are, of course, abundant in humans, some firmicutes that are abundant in humans. Again, we have natural genetic variation, so we have a lot of variation between our individual microbes. One interesting thing is that we have an awful lot of archaea that in our lab-raised juveniles. It'll be interesting to determine if this is because we were looking at juveniles or because archaea are actually doing better in the lab. And we ran some stats to show that indeed, looking at richness and evenness and beta diversity, that there's not much difference between these wild guts and our lab-raised guts. This is also kind of interesting because in zebrafish, we know that juvenile zebrafish tend to have a much more diverse microbiota than adults, so that it'll be interesting in our longitudinal studies to determine whether that's different in stickleback or whether we see more diversity in juveniles compared to adults in our lab-raised longitudinal studies. So in addition to looking at microbiota by 16S, we also like to play with microbes in the lab. So we've isolated a number of different microbes from the environment. This is a figure from a paper that we published in 2016 comparing the microbes that we've isolated from wild-caught marine and freshwater fish versus lab-raised marine and freshwater fish, showing that there were microbes that were present in the wild that were not present in the lab-raised fish in Oregon. So this is why we did such a careful study in our Alaskan population to make sure that we do have a similar microbiota between our wild and our lab-raised, but showing that there were a large number of microbes that we were able to isolate from the guts that were found in both wild and both wild and lab-raised fish. And because we're isolating microbes, we actually have a really nice collection now. We have over 280 microbial isolates at this point. They represent 4 phyla, 22 genera, and a number of unidentified microbes. The microbes were isolated from fish ranging from 14 days post-fertilization, which are those larvae or juveniles, all the way through adults. And we actually have both wild and lab-raised. We have isolates from both wild and lab-raised fish from both Oregon and Alaska. We have over 100 isolates now that are from Alaska. And we're doing a bunch of comparisons between those microbes. Now, the microbiota characterization that we've done so far is from wild-caught, and we really like to determine what the host genetic background contribution is to the difference in the gut microbiota. If we are grabbing these fish from the wild, they're raised in very different environments. So to determine what the host genetic background contribution is to these microbial communities, we're going to control the environment that these fish develop in. So this is showing a common garden experiment that we did in the Cresco lab when I was a postdoc, and I did this with Clay Small, in which we raised different populations of stickleback all in the same body of water. And this is something that we're going to be doing here at the University of Alaska Anchorage. And this will help us determine whether the host genetic background is selecting for different microbes, or is it that the fish in the wild are just exposed to different microbes, and that's why they have different microbial communities. So this is an experiment that we'll be doing here in Alaska with several different freshwater and anadromous marine populations from our populations here in Alaska. Okay, so that's a contribution of the host genetic background to microbial membership. Now we're going to move on to the immune response to microbes. So as I said, we're able to do some fun things with stickleback. We have a bunch of different microbial isolates. I'm just showing here that we were able to fluorescently tag some of those microbes and put them into fish, and we were able to determine that our microbes are actually colonizing fish and our juvenile fish. So this is a 14-day post-fertilization fish. We have a fluorescently tagged microbe that you can see here in the gut of these fish. And we were able to use this and a number of other methods to determine that the fish gut is colonized at 12 days post-fertilization. So for our experiments, we carried out experiments at 14 days post-fertilization. This is a 14-day post-fertilization fish. You can see that the yolk sac that you might have noticed in an earlier image, it was a big blob of yellow that was underneath here. That's totally gone in 14-day post-fertilization fish. So this fish is now surviving on food that it gets from the environment. Since we don't have a germ-free fish food yet for stickleback, we can only raise our fish to 14 days post-fertilization. And so we're terminating our experiments at 14 days. But at this point, we know that the gut is colonized. As a postdoc, I worked with Erica Mitke, who was a lab tech in Karen Gilliman's lab, to establish an assay in which we're able to count neutrophils. And I mentioned that earlier. This is just showing a cross-section of a fish. We look at the neutrophils that are here in the gut. This is the neutrophils that were present in a 14-day post-fertilization marine gut, or oceanic. And this is a conventionally raised fish. When we raise this fish in the absence of microbes, there are significantly fewer neutrophils present in this gut. And we're able to go through all these sections and count the number of neutrophils to determine how many neutrophils are in the gut. So when we look at our oceanic or our marine fish, when they're raised conventionally in the presence of microbes, we see a large number of neutrophils in the gut, somewhere between 100 and 200 microbes per fish. Each one of these dots represents the number of neutrophils in an individual fish, and this is the average and the variance. When we raise these fish in the absence of microbes, so germ-free, we see far fewer neutrophils present in the gut. When we monoassociate, in this case with that Pseudomonas strain that you saw earlier, we see an intermediate number of neutrophils in the gut, indicating that this particular Pseudomonas is able to recapitulate some of the immune response to microbes in the gut, but not fully. And so there are other microbes that are likely responsible for this large number of neutrophils in the gut. So that's exciting in and of itself. We have an immune response to microbes in this population of stickleback, this oceanic or anadromous population of stickleback. But we're really interested in how the host genetic background contributes to the number of neutrophils in the gut. So we wanted to look at a freshwater population. And I wouldn't show you this data if we didn't see anything interesting. So obviously we found something interesting. And what we found is that the number of neutrophils in the gut in our freshwater fish did not respond to changes in the microbiota. So here we have our conventional freshwater fish versus our germ-free. In oceanic, we had a big difference. In our freshwater population, we did not have a big difference. And our monoassociated looked very similar to our conventional and our germ-free. So this is exciting. Now we have evidence that the host genetic background contributes to the immune response to microbes in the gut. This is just for one population of stickleback. These are Alaskan populations. This oceanic is a rabbit slough, and this freshwater is Boot Lake. So this is a lake here in Alaska that we can sample from, and an anadromous population that we've already started sampling from. But this is just for one oceanic and freshwater pair. What we're really interested in now is determining whether this immune response is observed in other populations as well. So we've gone out this summer, we went out and grabbed fish from 2 different freshwater and 1 anadromous population that are different than these. We did some crosses, raised them germ-free, and exposed them, raised them either germ-free or conventional, and we're repeating these assays with the new populations that we're working with here in Alaska. So the next thing that we're interested in, the next thing I'm going to talk about is whether the host genetic background contributes to the ability of the host to protect against disruption of the microbiota by antibiotics. And the reason that we're interested in this is because we know that antibiotics affect the diversity of the microbiota. So this is a paper showing in mice that the diversity of microbiota decreases when you expose mice to vancomycin or to other types of antibiotics. And in this particular figure, they're showing that different antibiotics affect the microbiota differently. And they're also showing that this difference in the response to antibiotics, or this exposing these animals to antibiotics, also changes the number of C. difficile specifically that are in these hosts. So what we're interested in is determining whether or not the host genetic background can protect the host from changes in the gut microbiota. So we have 2 experiments that we're carrying out. We have a number of different experiments, but I'm going to highlight 2. One is funded by an NSF postdoc grant for Dr. Emily Leskak, who is a fabulous postdoc in my lab. And this NSF proposal focuses on chronic exposure to low levels of antibiotics. And what we're hoping to do here is mimic the level of antibiotics that are observed in the wild. So when we take antibiotics and we don't metabolize all of the antibiotics that we take as humans, and at the end, some of those antibiotics end up in our wastewater. The wastewater treatment right now does not get rid of all those antibiotics, and so now we have low levels of antibiotics that are constantly present in the environment, and we have fish that are evolving in the presence of these low levels of antibiotics, and they're developing in the presence of these low level of antibiotics. And so Dr. Leskak's project will focus on those low level of antibiotics and determine how that's affecting the evolution and the development of the fish in the presence of these antibiotics. And then we have our master's student, Ryan Lucas, who's working with Dr. Leskak on the low level of antibiotic exposure, but he also has his own funding through Embry to look at the short-term clinically relevant levels that are similar to the levels that kids are exposed to early in development. So what Ryan is looking at is a higher concentration of antibiotics, and how does that affect the development of the fish, not the evolution, but the development of the fish. And what we're doing here is we're doing short exposures of high concentration of antibiotics to mimic what kids are exposed to. So for example, if you take your kid in to the doctor for an ear infection, and the ear infection is treated with high levels of antibiotics, and you do that once or twice or 3 times during the child's development, how does that affect their development? And how does the host genetic background protect your child or not protect your child against the disruption of their microbiota due to this short-term antibiotic exposure? So we have an experiment that we're hoping to start this summer, a long-term experiment in which we expose fish to antibiotics, low levels or high levels of concentrations of antibiotics for up to a year. And this will help us determine whether these fish are affected by, whether their development is affected by exposure to antibiotics. And we'll do this with a couple of different populations, oceanic and freshwater populations of fish, to determine whether the host genetic background affects the development, affects the both the microbiota community structure and the immune response to microbes and immune development in these different populations of fish. And we're also looking at their overall development. So that's a long-term experiment. But to determine whether or not these low level and high level of antibiotics are able to affect fish, we also did a short-term experiment. So we went out to a couple of different populations, Big Lake, Mud Lake, and Westchester Lagoon. This Mud Lake has anadromous fish in it. Even though it's a freshwater lake, it has anadromous fish that come in from the ocean, come into the ocean, in from the ocean, and they lay their eggs in a freshwater mud lake, and then they go back out to the ocean. Big Lake is a freshwater population, and Westchester Lagoon has both anadromous and freshwater populations. But in this particular experiment, we focused on the freshwater fish in Westchester Lagoon. So we did a short-term exposure to 2 freshwater populations and 1 anadromous population. We fertilized eggs at 0 days post-fertilization. The gut opens about 10 days post-fertilization. We added antibiotics, so we disrupted their gut microbiota at 12 days and collected the fish at 14 days. We measured health and development of the fish by looking at somatic development. So this is just what the fish look like as from the outside. What can we measure overall in their overall development? We also looked at behavior. And in progress right now is characterizing the gut microbiota, and we'll be looking both at diversity and membership. So how is diversity affected by the antibiotic exposure, and which microbes are actually killed off by the antibiotics, and which are able to survive antibiotic challenge. We did this in triplicate. We exposed fish to either no antibiotic, the solvent ethanol, which is the solvent for the antibiotic, in either low or high concentrations of tetracycline. And then we did this in triplicate for our 2 freshwater and our oceanic population. And we did them, we raised them in these flasks. And here's the timeline once again. We looked at behavior by putting these flasks on a grid and looking at the movement of individual fish. So here's an individual fish on our grid, and we looked at the total number of squares that these fish moved through, the number of novel squares that they moved into. So did they just go back and forth between these 2 squares, or did they go exploring all over the flask? And then we looked at how long they spent on the perimeter, or how many perimeter squares they traversed into. So were they hanging out here in the center like these guys, or were they only staying on the perimeter? So for example, if this guy only went down here and across here, that would be a fish that was hanging out just in the perimeter. In our Big Lake freshwater population, we didn't see a difference. And so these measurements were taken by Haley Miller, an undergraduate that worked in our lab last summer. And we looked at low TET and high TET in the ethanol and the control samples. There was no difference in the distance traveled, the number of parameter squares that they went into, or the number, how long they spent on the parameter in terms of how many squares they went into, or the number of squares that they entered. So this was interesting in and of itself, because now we know that the host genetic background did not affect the behavior of the fish that were developing under antibiotic exposure versus control. But we also did gross morphological measurements. So this is just looking at their overall somatic development. We measured the length of the fish. We looked at the swim bladder length and the swim bladder area, because the swim bladder is important in the development of the fish. And then we looked at the eye diameter, and this is a measurement that's been used by other groups to just determine if there are differences in development from one population to another. And what we found is that when we looked at our oceanic population, and we looked at a measurement, in this case, standard length or overall length, there wasn't a difference between fish raised in control or ethanol treated, or in the presence of low concentrations of tetracycline or high concentrations of tetracycline. And again, similar to the other box whisker plots, individual dots represent the lengths of individual fish, and then these are the averages and deviation. However, when we looked at our big lake fish, which is the freshwater population, we saw a decrease in the standard length for our ethanol treated fish, and we saw a little bit of a difference in our high and low concentration fish compared to our high concentration of tetracycline compared to our control fish. However, this is a really small difference. It's not a huge difference, so we can't really say that this particular population is responding that much, but we looked at our other freshwater fish and we saw a huge difference. So this is our Westchester freshwater fish. The controls were significantly smaller than our oceanic and our freshwater big lake fish. And our antibiotic treated fish were very different in their size compared to the control. So here we have 3 populations with different genetic variation, with different genetic backgrounds. One freshwater population has a significant difference in their standard length when they're raised in the presence of antibiotics, but our oceanic and another freshwater population does not. So yay, we have something fun to work on for a while. And as I said, we did lots of other measurements, and this is just showing that the other measurements showed something similar, except for swim bladder area. And I can talk about that if y'all want to talk about that later. So our conclusion is that there's a gene by environment interaction that appears to drive the response to antibiotic exposure. The freshwater have increased developmental instability, and there's no influence on behavior, which is interesting in and of itself. And Emily will be following up on that. As I said, we have all these different microbes that we've isolated, so we've started playing with them, looking at the resistance to different antibiotics. Here I'm just showing TET and gentamicin, but we're also doing another antibiotic. These are really early in the studies, and so the numbers are really low here, but we're looking for differences in antibiotic resistance between our populations. We're also looking at acid tolerance. Ruth started this. Ruth, my former lab tech, who's now a PhD student at the University of Wisconsin-Madison, was looking at acid tolerance because there's a difference in the pH between these different bodies of water. And she found that while I'm only showing 8 strains here, but she's looked at more strains since then and found that there was a difference, and that the Westchester Lagoon, which is more acidic, is also, the microbes from that population are also more acid tolerant than Big Lake, which has a more neutral pH. We also started characterizing our microbes, both in terms of motility and biofilm formation. This is something that Rachel is now working on. This was started by Ruth, and now Rachel is looking at determining whether there's a difference in the microbes isolated from the guts of fish from these different populations in terms of motility and biofilm formation, because this will give us some indication of what's actually going on in the lab. And this is just showing how these assays are done. So, host genetic background. Does it contribute to microbiota membership? Possibly. We're working on getting more information. Does it contribute to the immune response to microbes? Yes, but we're working on determining whether that trend is generalizable across populations of Alaskan fish. And does the host genetic background protect against disruption of the microbiota by antibiotics? Yes, but we're confirming and working on a mechanism. So, future directions. We just got some funding, INBRI funding, to look at whether or not the host genetic background contributes to the stability of the microbiota, and whether when the microbiota is exposed to perchlorate. Perchlorate is an environmental contaminant that's found throughout the United States, but it's especially in high concentrations around military installations and places where explosions are set off on a regular basis, because it's a byproduct of those explosions. And we know that there are sites here in Alaska that have particularly high concentrations of perchlorate, and that this this perchlorate can become concentrated in fish and other animals that eat perchlorate contaminated fish. And Dr. von Hippel has found in the past that perchlorate affects the development of the fish overall, and that this is reversible with iodide, and this is due to perchlorate disrupting the iodide SIM transporter in the thymus. But when he treats these fish with iodide, he does not see a complete restoration of the health of these fish. And so we hypothesize that the gut microbiota is also being affected by perchlorate in a manner that's not directed by that iodide SIM transporter in the thymus. So we looked at the ability of gut microbiota isolates to grow in vitro in the presence of perchlorate. And we saw that, for example, in this Listonella, when we grow this microbe in the absence or just in the presence of instant ocean, that there's no effect of perchlorate on the growth of this, that this microbe grows just fine. But when we grow this microbe in the presence of perchlorate, we see a decrease in growth. So this particular gut isolate does not grow well in the presence of perchlorate. We've looked at 10 different strains, 17 different strains. 10 of those strains had growth that was affected. Some of them had increased growth, but most of them had decreased growth. And there were 7 strains that didn't, in which their growth was not affected by perchlorate, the presence of perchlorate at all. So we hypothesize that the gut microbiota is going to be affected by the presence of perchlorate. And so we're doing some experiments this summer, both short-term and long-term, to determine whether perchlorate disrupts the gut microbiota. So if you're a person and you're drinking perchlorate contaminated water, is that going to affect your gut microbiota? We'd like to know. So we're using stickleback as a model to study this. So we'll be determining whether perchlorate affects the gut microbiota, whether that's recoverable, whether that phenotype is reversible with iodine, and whether that affects the development of the host overall. And then the other thing we're interested in, I have a new postdoc that will be joining our lab at the end of August, Lucas Kirchman, who comes from Southern Illinois University. He's with Robin Warren's lab right now. He'll be joining our lab to look at whether the gut microbiota is affected by whether the host genetic background contributes to the gut microbiota differences, and whether that affects the ability of pathogens to colonize and affect the development of hosts. And this is based on work that has shown that when you disrupt the gut microbiota, that pathogens are then able to, are much more likely to be able to infect a host and cause disease. And I'm using the term pathogens broadly here just to indicate microbes that are actually able to cause, or that will cause disease in a host. And that's a shout out to Arturo Casadevall. I'm trying to use the term anyway. So in this particular case, we're interested in this question of, does the host genetic background influence the susceptibility to pathogens? And is this due to a difference in the microbiota, the gut microbiota? We know for other diseases, so for example, dengue virus, that the ability of this virus to cause disease in humans is due to the genotype of the virus itself, the environment that the human that the human is exposed to, and then whether and the host genetic background. So this is a gene by gene environment, gene by environment interaction in which the ability of this virus to cause disease in humans is based on the genotype of the virus, the genotype of the human, and the environmental differences. And so we're interested in exploring this idea in stickleback. And so we'll be challenging conventional germ-free and mono-associated fish from different populations with different pathogens, starting out with known pathogens like E. tarda or Vibrio, and then identifying key differences in survival, pathology, and inflammation between these populations. That's it. That's all I'm going to talk about today. We have other things that are going on in the lab, believe it or not, a lot of other things that I didn't have time to chat about. my lab, I've been very lucky to have a big lab with lots of great people. So the people in gray with the stars are people who have been in my lab in the past, who've moved on to bigger and better things. And the people in white here are the people who are currently in my lab. Coral, Kelly, Rachel, Jeremiah, and Kenny are all undergrads who are doing graduate level work because they're so independent and developing their own assays. Ryan is a master's student who is making great progress on his work with our postdoc, Dr. Emily Leskak, and I talked about her work in quite detail today. At UAA, I've been really fortunate to work with Brandon Briggs and Janelle Skiskorsky. They have helped me a lot with looking at the comparing the microbes in the water to the microbes in the gut. And of course, we have our perchlorate experiment with Frank von Hippel, who is now at Northern Arizona University. That we'll be continuing this collaboration for quite a while. And Frank has just been a really wealth of information in terms of just talking about stickleback and stickleback development and et cetera. We have funding. We've been very lucky to have funding from NIH. We have an R15 that's supporting the funding of the comparison of the gut microbiota in different populations. We have funding from INBRI and 2 pilot studies from INBRI. Dr. Leskak has an NSF postdoctoral grant. American Fisheries Society supported our high school students and has supported, and UAA has been very generous in supporting my research as well. And a lot of this work was started at the University of Oregon with some fabulous, talented undergrads. You should be looking for these people because they have moved my projects forward immensely. Magna and Marianne and Patrick are all pre-meds. Patrick is already almost done with med school, and Marianne and Magna will be starting med school soon. You should be looking for their applications because they are fabulous students. The Gilliman Lab, Karen was a fabulous mentor. Erica and Travis helped immensely with assay development. Zach Stevens helped me immensely with gut microbiota characterization. Heather fluorescently tagged my microbes for me. And then I also worked with some folks in Bill Cresco's lab. Mark Curry, who taught me everything I know about stickleback fertilization and development, and Clayton Small, who we just published a paper earlier this year or last year on the transcriptome of germ-free and conventional fish. All right, that's it. Hopefully some of you are still there.
Thank you for a very comprehensive talk.
Yeah, it was a lot.
I did tweet, I was waiting for you to show the three-eyed fish from Blinky after the antibiotic treatments. The Simpsons.
Oh man, I'm going to add that to next year's talk.
So I think Joe's the only one left in the Hangout. Do you have any questions, Joe? I can unmute you. How do I unmute him? Hold on a second. And yeah, if there's any questions on Twitter, please let me know. Use the hashtag or tweet to microseminar. So I do have a question for you. I know we had talked about this previously, but when you showed the wild versus freshwater gut microbiota being so different, we had talked about sort of time since feeding and like quality of like, how did you dissect that? Is that poop, or do you have an idea of how long it had been since they all ate?
Yeah. So for both the oceanic and the freshwater fish, they were collected and then brought to a spot where we were doing the gut dissection. So they were in buckets of water for a couple of hours before we did the gut dissection. So I don't know exactly when they lost 8 in the wild, but we tried to control a little bit for that time difference between collection and gut dissections. So in our lab studies, we'll be able to regulate that tighter. We'll be able to, feed them and then wait 24 hours and then do the gut microbiota collection so that we're not getting their food in the microbiota sample.
And you mentioned something about germ-free food.
Yeah, I'm working on that.
Can you not believe it?
Yeah, so we have, as a postdoc in Oregon, 4 of the years I spent was I had 5 projects going on in Oregon, and one of those was developing a germ-free food source for zebrafish. And so I'm taking that knowledge here and developing a germ-free food source for stickleback. It's one of the other side projects that I have going on, where I think it'll be pretty straightforward in stickleback because we can make their shrimp germ-free pretty easily. So that's something that we're working on this summer, possibly later today, but maybe next week.
All right. So I don't see any other questions coming in. Are there any questions? I can't figure out how to get back to the chat window. Any questions by chat? No. Okay, well, with that, thank you so much, Kat. And we will have this archived on YouTube, and there will be another seminar headed to you next month.
All right. Thank you so much, Jen, for inviting me.